What is protein synthesis? Protein synthesis is a process in which proteins are produced. This process will happen with the help of DNA and RNA. What is DNA? It is a double helix strand with 4 nitrogenous bases. What is RNA? It is a nucleic acid that functions in protein synthesis. Before we proceed, let us unlock some difficult terms that will help you understand protein synthesis. Amino acid, the building blocks of proteins. There are 20 different types. Codon, a sequence of 3 organic bases in a nucleic acid that code for a specific amino acid. Exon, coding region of eukaryotic gene, parts of the gene that are expressed. Gene, a length of DNA made up of a number of codons, codes for specific proteins. Intron, non-coding region of a gene that separates exons. Polypeptide, a chain of amino acids joined by a peptide bond. Ribosome, a cellular organelle that functions as a protein-making workbench. RNA, ribonucleic acid, a nucleic acid that acts as a messenger. At this juncture, let us picture out how protein synthesis looks like. Protein synthesis has 2 processes, the transcription and translation. In transcription, there are 2 sub-processes, and in translation, there are 3 sub-processes. In transcription, there is initiation and elongation. On the other hand, translation has initiation, elongation, and termination. Now let us go to transcription. The first phase is the initiation. The DNA has been prepared inside the nucleus. We can see in the drawing is the cell with its organelles, especially the ribosome. Afterwards, the RNA polymerase has been prepared. The RNA polymerase unzips or unwinds the DNA. Then the DNA produces a copy that eventually will be a single strand. Then it will be transformed into an mRNA. In DNA, there are 4 nitrogenous bases, the thymine, guanine, adenine, and cytosine. In mRNA, there will be no longer thymine, and it will be changed into uracil.
Next phase is the elongation. What will happen in this phase is the mRNA goes out from the nucleus and travels going into the ribosome to start the translation process. Now let us go on with translation process. When the mRNA reaches the ribosome, the translation process will start. Ribosome attaches to the mRNA at the start codon. Translation of mRNA into the amino acid language of proteins require an interpreter. To convert the codon to the one-letter amino acids word of proteins, a cell employs a special molecular interpreter, which is called transfer RNA or tRNA. Each tRNA is bonded to a specific amino acid. The next phase is the elongation. The next amino acid, tRNA, attaches to the adjacent mRNA codon. The bond holding the tRNA and amino acid breaks down and polypeptide bond is formed between the adjacent amino acids. As the ribosome can only cover 2 codons at a time, it must now shuffle down to cover a new codon. This releases the first tRNA, which is now free to collect amino acid. The steps will repeat again. Now we will go to the termination phase. As the polypeptide chain elongates, later on it will peel away from the ribosome. But before that, elongation continues, perhaps for thousands of amino acids, until the ribosome reaches one of the 3 stop codon. At this point, the mRNA dissociates from the ribosome. Translation is complete.
To sum all up, before transcription, the DNA is being prepared in the nucleus of the cell. Then the DNA enters into the RNA polymerase, and the DNA makes a copy of its own, but it will be now a single strand. The thymine will change into uracil, and the single strand will now be called as mRNA. This is how the process transcription works. The process of translation starts when mRNA binds into the ribosome. The codon starts to fill the sites, and amino acids will grow a polypeptide chain. The bond holding tRNA and amino acids break down, and a polypeptide chain will be formed, and the tRNA is free to collect another amino acids, and the process continues.